All right, so welcome to week 5 of microbiology. Congratulations to all of you for surviving the first exam. So all of the exams should be graded by now, and your grade should be available on Blackboard. If you're a little weary about your grade result, at least you guys now are aware of the type of format that you should expect for future exams. So hopefully you will be able to prepare more effectively and efficiently for the next upcoming exams. So we are going to move on now to chapters 5 and 23. Both of these are talking about eukaryotic cells. So parasites are a particular category of eukaryotes consisting of the protozoa, helminths, and the ectoparasites, which are insects that actually can spread disease. Now, for chapter 5 focuses on basic characteristics of all eukaryotic cells. So we've introduced kind of the foundation of the two cell types that exist in nature, this less complex prokaryotic cell, which we know archaea and bacteria are prokaryotes, and then all of the eukaryotes, which for the purposes of microbiology include fungi, algae, protozoa, helminths. So I'm going to jump right into lecture with introducing this idea of the origin of a eukaryotic cell. So this is definitely a theory that you should be aware of, and this is what is known as the endosymbiotic theory or endosymbiosis. And essentially, this theory suggests that organelles originated from prokaryotic cells that were trapped inside of them. And so this idea of a eukaryotic cell is the result of a larger cell engulfing a prokaryotic cell to form this symbiotic relationship, which we now call a eukaryotic cell. And why do we think that this is the case? Well, if you actually look at, for example, these little guys here, this smaller prokaryotic cell, it's saying here, similar to purple bacteria that can use oxygen. So here you see this larger cell engulfing this smaller prokaryotic cell. What do we think this is here now in our modern day, even our cells, our human eukaryotic cells? This is actually a mitochondrion, and mitochondria are very closely similar structurally and functionally to the garden variety bacterial cell. They have their own genome, they have a circular chromosome, they carry out their own metabolism. So this is basically, you see this described as the powerhouse of the cell. This is the site of where the majority of ATP or energy is made for cells to drive all of their processes, their biological processes. So it has its own DNA, it has its own metabolism, it has its own membrane. So this is kind of evidence suggesting that this larger cell kind of engulfed this smaller prokaryotic cell and over time formed this symbiotic relationship. Now, of all of the eukaryotic microbes that exist that we will be talking about this semester, they are the following: protozoa, which always are unicellular; fungi and algae, which could be either unicellular, this would include the yeasts, or multicellular; and the helminths, which are always multicellular except for the reproductive stages. So there's 3 stages that characterize the life cycle of a helminth or a parasitic worm. That would be an egg transitioning to a larva, transitioning to the adult helminth worm. So this is your average cartoon image of a typical eukaryotic cell, and we're going to go over all of the specific components of a eukaryotic cell. So this would include the envelope, so potentially a cell wall, and always a cell membrane. Also, we're going to see a lot of new structures popping up with eukaryotes that we did not see in prokaryotes, and you will see some overlap. So generally speaking, eukaryotes are larger. They are indeed more complex. They are more organized in the sense that they have these specialized compartments here, these specialized functional compartments, the mitochondrion, for example, which will produce the majority of ATP, or adenosine triphosphate, which can be used to drive chemical reactions. Also, the Golgi apparatus, the rough and smooth ER, which sits adjacent to the nucleus, which is the most prominent organelle, which houses the genetic material. Also microtubules, and there's multiple types of filaments or scaffolding that you see internal to cells. If you don't have a mitochondrion in animal cells, and rather you are a photosynthetic cell, you may see chloroplasts, also centrioles, which will be used during cell division. And I forgot the nucleolus, which is within the nucleus, which is basically just a specialized site of the nucleus where you would find all of the ribosomal DNA. And also along the outer shell of a eukaryotic cell, as we saw with prokaryotes, I mentioned this component that we call glycocalyx. So this is that slime layer, which is also overlapping with eukaryotes. And also flagella. And with eukaryotes, they may additionally have cilia, which we do not see in a prokaryotic cell. So I'm going to kind of split up the characteristics of an average eukaryotic cell into the external structures, the boundary or the envelope, and then we'll go through all of the internal contents. So external to a eukaryotic cell, you may find flagella, which we saw in prokaryotes as well. And what I would like to point out here is that typically a eukaryotic flagellum is thicker. So it's 10 times thicker generally. And same functionality, it functions in motility. Now, I should note here that not all eukaryotic cells have all of these structures. So you won't see a flagella on every single eukaryotic cell, more specific to prokaryotes than eukaryotes. But cilia are unique to eukaryotes, and they are found only on a single group of protozoa and certain animal cells, like the cells within our respiratory epithelial lining. So these structures can function in not only motility, potentially feeding, but also filtering. So the whipping motion of these cilia can serve all of these functions. And similar to what we saw in prokaryotes, eukaryotic cells may also have this outermost layer that typically is composed of polysaccharides and can be, remember, loosely or more tightly organized layer, so a slime layer or capsule. Same functions here, functions in adherence, protection, signal reception. And specifics here unique to fungi. Most fungi and also algae have a thick, rigid cell wall, whereas the majority of other eukaryotic cells do not have a cell wall and only have a cell membrane. So all cells definitely will have a membrane, but not all eukaryotic cells have that cell wall. And if they do, it's made up of different structural units than we saw in prokaryotes. So remember, with prokaryotes, you either had peptidoglycan as that repeating framework of NAGs and NAMs, or we saw mycolic acid. Here, if eukaryotes have a cell wall, if it's a fungus, they have this thick inner layer. It's polysaccharides again, but it's this chitin component. And algae may have cellulose, pectin, mannans, silicon dioxide, and also possibly calcium carbonate as part of their cell wall. Now, every eukaryotic cell must have a cell membrane. So all prokaryotes and eukaryotes, no matter what type of microbe, definitely has a cell membrane that serves as that selectively permeable layer. And also, within a eukaryotic cell, they have all of these specialized compartments that we call organelles that account for 60 to 80%, the majority of their cell volume. And what's key to remember here is that these organelles themselves have their own phospholipid layer, so they have their own membrane. So the nucleus is a perfect example of this. It's the most prominent organelle that you find within any eukaryotic cell. And surrounding the nucleus is the nuclear envelope, which you see these little dots here. These are nuclear pores, and the pores are basically serving as these regulated compartments through which specific larger molecules can enter or exit. And so there's a regulated mechanism through these pores for what is being transported in and out of the nucleus. And this is critical because residing within this prominent organelle is the software of life. It's the code, it's the DNA, right? So the chromosomes that you find within the nucleus contain all those A's, T's, C's, and G's, adenine, thymine, cytosine, and guanine. Every organism has its own genome, and all of that information basically tells the cell how to carry out all of its biological processes, how to develop, how to carry out any process that it needs to thrive. And then there's this highly dense specific region within the nucleus, which is pointed out here, the nucleolus, and this is specific for ribosomal RNA synthesis and ribosome assembly. So a ribosome is the machine that is used to make proteins, and ribosomal RNA is actually part of the ribosome itself. Now, important to mention here, if you are a eukaryotic cell, the genetic material in the form of chromosomes is typically linear. If you are a prokaryotic cell, usually the chromosome is in the form of a circle. So it's just a single circular piece of DNA that's essential in a prokaryote. If that prokaryote does not require a specific piece of DNA, the extra piece of DNA may be called a plasmid, but the essential piece of DNA is always called the chromosome. Now, because we are talking about eukaryotic cells, just going back to prokaryotes, we said that they divide via binary fission. For a eukaryotic cell, it will divide via mitosis. And so what happens during mitosis is this is then essentially M phase of the cell cycle. So there's 4 stages of the cell cycle. There's G1, S, G2, and M. So all of these steps are within M, which is referring to mitosis. And during mitosis, essentially, the DNA will condense. So this is following DNA replication. The DNA condenses and it undergoes the following phases here from interphase. So this is the resting state before it gears up to divide. Prophase, where you have the chromosomes that have been duplicated and the centromeres, spindle fibers are actually moving to opposite sides of the cell. The centromeres are actually the middle parts of the chromosome. During metaphase and late metaphase here, all of the chromosomes are actually lining up on what we call the metaphase plate. And then this transitions into anaphase, which is characterized by these sister chromatids actually pulling apart. So this is basically each one of these here is a replicated chromosome. So each sister chromatid here is identical. So the idea is after the DNA has been replicated, then these chromosomes are going to be pulled apart during anaphase. And then finally it progresses into telophase, where you start to form this cleavage furrow. And at this stage, the cell will undergo the process known as cytokinesis. Ribosome is the macromolecule that will basically take RNA and convert it to protein. So the proteins are synthesized here, and then they are sent to the smooth ER for packaging and transport. The smooth ER, again, it does not have ribosomes, and it can function additionally in nutrient processing, synthesis, and also storage of lipids. So just remember, rough ER you can always tell because you see, even in a cartoon image, it's riddled with these dots, which are representative of ribosomes. And this one single dot here is representative of this whole structure. So if this is a ribosome, this is the small subunit, this is the large subunit, this is where that RNA template is fed through. It's actually the messenger RNA template. And usually there's multiple ribosomes that read one mRNA template. So whatever the code is in this sequence, so in DNA, it's A's, T's, C's, and G's. In mRNA, it's A's, U's, C's, and G's. And so the ribosome reads all the A's, U's, C's, and G's in the form of a triplet code. So it will read it in groups of 3, AUG, CCG, CCA, for example. And according to what that triplet is, it will make the associated amino acids. So these little green dots are the amino acids. And once the ribosome actually reads this mRNA chain, it will link multiple amino acids together, making this single polypeptide chain that we call a protein. So this basically goes on. Once it's released from the ribosome, it will then fold into the associated protein. So also we have the Golgi apparatus, and the Golgi is usually adjacent to the ER, and this is where proteins are modified, also packaged, and they can be shipped off into vesicles. And the specific membranous sacs that make up this Golgi apparatus are referred to as cisternae. Now, I would like us all to remember kind of the general pathway here for transporting proteins that start off here. In order to actually express that protein, you need to start off in the nucleus. And then how does that protein actually get shipped to outside of the cell or extracellularly? And so the pathway here is that if you want to actually express a protein, modify it, package it, and ship it out of the cell, we have to start off in the nucleus where the chromosomes are, where the DNA, the sequence, A's, T's, C's, and G's are. You need to read that gene, so express that gene to make RNA, and then the RNA is going to be shipped out of the nucleus into the rough ER. And remember what's specific about the rough ER. The rough ER has ribosomes, right? So the mRNA is going to make contact with the ribosome, which is going to read the sequence A, U, C's, and G's. It's going to make the protein. The protein is going to be sent to the Golgi apparatus, modified, packaged into a vesicle, and shipped outside the cell. So I would remember this pathway, starting off with the sequence of DNA in the nucleus. That gene is expressed, or it undergoes this process that we call, in other words, transcription. The RNA is then shipped to the rough ER where it meets the ribosomes, which will translate the RNA to make protein, then shipped to the Golgi, packaged in a vesicle, and secreted outside of the cell. Now, there's also some additional internal structures, such as lysosomes, vacuoles, and phagosomes. Lysosomes are vesicles containing enzymes that come from the Golgi, and these guys basically are garbage disposal, so they're involved in intracellular digestion of extra food particles, and they also serve an antimicrobial function, so they protect against invading microbes. A vacuole basically contains components that will be fated for storage or even digestion or shipped out of the cell. And a phagosome is basically these guys merge, so a vacuole merged with a lysosome. And then we have the mitochondria. Remember, this is the reason, the rationale behind why a lot of scientists agree with this endosymbiotic theory. So it's the mitochondria and the chloroplast that is structurally and functionally analogous or very similar to a prokaryotic cell. So remember, if we're talking about a bacillus, it's a rod-shaped bacterium, which looks very similar to a mitochondrion or a chloroplast. It has its own membrane, so the cristae here internally, and even this outer membrane as well. It actually has its own ribosomes. And these are, go figure, very similar to prokaryotic ribosomes. It also has its own circular piece of DNA, or its own circular chromosome, so that it has almost all of the structural characteristics of a single bacterial cell. Now the key here is that the cristae, this inner membrane, holds all of the electron carriers that function in aerobic respiration. So without going through the whole process of the electron transport chain, let's just suffice to say that essentially these electron carriers here will funnel electrons through a series of what we call cytochrome or electron carriers, and in the end, what happens is there's a buildup of protons on one side of the membrane, and through this chemiosmotic gradient, so it's a chemical gradient, so instead of water, there's a buildup of protons on one side of the gradient, one side of the membrane. They diffuse through the other side, and they spin through this enzyme called ATP synthase, which makes ATP. So what should you remember here? This mitochondrion is going to make a ton of ATP. And they also contain their own DNA and also their own ribosomes. Now, chloroplast, this structure, this organelle, very similar to a mitochondrion, except some of the processes are flipped here. So instead of aerobic respiration, we have these chloroplasts, which are essential for this process that we call photosynthesis. So they are able to take sunlight and convert it into chemical energy. So they produce oxygen and also glucose. And these are found in algae, also plant cells, and rather than in here, we saw these cristae. They contain these thylakoid membranes stacked in these units that we called grana, and together a granum. And they are cells that contain these organelles, are the primary producers of organic nutrients for other organisms. Now the ribosome, remember what I said about this structure? It is a macromolecule that again is the protein synthesizing machine. So we have a small subunit, a large subunit, and together what do you find within the small and large subunit? Ribosomal RNA and proteins. So even though it actually is carrying out a chemical reaction, it is not just solely made of protein, it's also made up of RNA. And these guys are scattered throughout the cytoplasm or also associated with the rough ER. And the thing to remember is that they're larger than those found in prokaryotes. So prokaryotes have them as well, and they serve the same function. So they're going to make proteins. Another important structure, all of the filaments that make up the scaffolding that we call the cytoskeleton. So the protein microfilaments and microtubules, they essentially form this scaffolding network which provides structure and also transport for molecules throughout cells. And there are these really neat little motor proteins that, if any of you guys have seen the inner life of the cell, you can look it up on YouTube. There are these little motor proteins that actually, through ATPase activity, they actually walk along some of these microtubules transporting cargo. It's really cool to watch, so check it out if you get a chance. All right, so now I'm going to talk about just some specifics, kind of zooming in on each of the eukaryotic groups that are considered microscopic. So there are some macroscopic fungi, but also the unicellular fungi, for example, yeasts. So the causative agent of a yeast infection, Candida albicans, is a microscopic unicellular fungus. Also, if you actually look at multicellular yeast or a mass of specific fungus growing on, let's say, mold on old bread that you left out on the counter, you will actually see microscopically on the cellular level that there's hyphae and spores. So there's smaller structures that we do need to mention. And then also algae, which all of these guys are photosynthetic. Protozoa, which a lot of them have these complex life cycles, which are very interesting and fascinating. And then parasitic worms, which are multicellular animals. They actually have their own primitive primordial organ systems. Some of them have sensory, digestive, reproductive systems, but they're not fully considered animals. They're multicellular, but also they have these microscopic life stages. So a little bit about fungi. There are both macro and microscopic, and there's actually 100,000 species. The majority of them are actually unicellular, and for the online group, you guys will be actually growing some fungi with one of the upcoming labs, and you'll be assessing growth conditions which are optimal for mold growth. Now there's microscopic fungi, which I mentioned are the yeast. So there's actually two morphologies here for microscopic. You may see yeast, which would be Saccharomyces cerevisiae, budding yeast, if anyone likes to enjoy a glass of wine or beer. Both of those are beverages that are crafted from fermenting various sources of starch, and the organism that will do the fermenting are yeasts. So these guys are what you see down here, the budding yeast. They're round, they have this ovoid shape, and they have asexual reproduction via budding. They also can undergo meiosis if they're diploid. And then on the right-hand side, you see hyphae. So this is for one of the structures you see in long filamentous fungi or mold. So when you see that massive furry stuff growing on bread, the smaller units are the hyphae. And then sometimes you see this unique growth characteristic where they may exist in both or either form. So this would be dimorphic, and usually this occurs when you have the yeast or the specific fungus that's growing in higher temperatures. So usually this is typically seen in pathogenic mold. So fungi that can use this to their advantage, they will exhibit this thermal dimorphism. Now, actually, the majority of fungi are harmless, and they're saprobic, meaning that they live off of dead plants and animals, but some can cause disease. So some are parasitic, and if they do cause a fungal infection, this is what we call a mycosis. Mycoses are multiple fungal infections, and they're extremely widespread. And there are many fungal infections, for example, that we see unique to certain geographical areas based on kind of what gets dispersed in the air from the soil environment. And usually the most severe fungal diseases are those that enter the lungs and then can become systemic. And these are generally more difficult to treat because they're eukaryotic based rather than prokaryotic. So since our cells are eukaryotic as well, it's trickier to find structures that we can target. Now for yeasts, we said that they have this oval shape. Now this is kind of a cartoon image here showing you all of the components in this little bud. So usually, for example, Saccharomyces cerevisiae will divide via this budding process. So this is the mother cell. The newly formed daughter cell usually buds off, and it's kind of cool. You can tell how old a specific mother yeast cell is by how many bud scars that the cell has. And this is an asexual process, this budding. Now you may see a multicellular mass of filamentous fungi. So the mass of hyphae growing on, let's say, a surface of an agar plate that looks like a furry sweater is called a mycelium. So this usually is cottony, hairy, or velvety texture. And if you take some of this mycelium and look at it under the microscope, you will see the smaller cells, which we call hyphae. And there's different types of hyphae. So hyphae can have septa or they can be non-septate. Another word for non-septate here is coenocytic. So if you see they have these septa here, usually there's pores actually in between the septa. And you may see some cases of vegetative hyphae, which will digest and absorb nutrients, or reproductive hyphae, which are different because they may produce spores, which are another component of some fungal species that can be used for reproduction. So this is not what we think of as an endospore for bacterial cells. It actually can be used for reproduction, where endospore is not. It's just a dormant cell type. Now, usually for reproduction, it's primarily through spores, and these are formed on reproductive hyphae. In asexual reproduction, the spores are formed through budding or mitosis. And there's two different types of spores. So you can have conidia or sporangiospores. And I would remember that the sporangiospore is kind of enclosed in a sac-like structure, whereas the conidia are kind of exposed freely on the surface of that spore aggregation. So those spores that I mentioned before, the sporangiospore you see here enclosed in a membrane-like structure versus the conidiospores that are exposed on the surface. And there's all different types of conidiospores depending upon the phyla of fungi that we're talking about. But both sporangio and conidiospores are types of asexual mold spores. Now, fungi can produce via sexual reproduction, which are different types of spores, whereby you have a fusion of two different strains in the formation of a sexual structure in the case of a zygo, asco, or basidio-spore. And so I would remember the following names. So if you look at kingdom Eumycota, there are several phyla within this kingdom based on the type of spore. So it's pretty easy to actually remember the associated phylum with the spores because they're, the name is very close. So phylum Zygomycota has zygospores, Ascomycota, ascospores, Basidiomycota, basidiospores, Chytridomycota, flagellated spores, and there's actually the imperfect group that produce only asexual spores. So if you look across the whole spectrum of fungi, again, the majority of them are actually harmless, but there are some that can cause nasty disease in humans and animals. And as far as identifying different types of fungi, it's based on the following characteristics. So the type of spore forming structures and spores, the type of hyphae, the colony texture and pigmentation as well. So how does it actually grow on an agar plate? Biochemically, how does it behave? And also, most definitively always would be the genetic makeup. So this would be based on ribosomal DNA sequencing. Now, as far as the role of fungi, like I said, the majority are saprobic, but some people actually have allergies. So some people are allergic to different strains of molds, or as we mentioned, they can cause disease. They also release these toxins called aflatoxins in some cases, and blight, for example, has destroyed potato crops. One of the reasons why this whole movement of GMO has started is to essentially allow for these pest-resistant or microbial-resistant crops that will allow for ensuring that the food actually is available. But let's not forget that Penicillium was the producer of the first ever discovered antibiotic. So as you see in the agar plate on the right here, this clearing around the mold. So this is all bacteria. Penicillin was produced by Penicillium. So a lot of fungi we find make naturally produced antimicrobial compounds that we call antibiotics. Also alcohol, so sake or Japanese wine is fermented rice, and this is from Aspergillus, which is a strain of fungus that will produce the ethyl alcohol. Also, a multitude of different acids and vitamins, and they're very easy to manipulate genetically in the lab. So Saccharomyces cerevisiae is a favorite to use in the laboratory environment. Now, there are a bunch that can cause diseases. Ringworm, so tinea, athlete's foot, also toenails, a lot of fungal infection of the nails, or candidiasis, yeast infection, especially for women, or oral thrush for late stage HIV and AIDS patients. And then there's more systemic, deep spreading fungal infections that can start off in the lung and spread, so these are more difficult to treat. A lot of these we don't see around here, histoplasmosis, for example, but it is good to know that there are a few fungi that actually can result in mycosis and are more challenging to treat. Now, as far as the protists, I would remember algae and protozoa fall under this umbrella term that we call a protist. Algae, they're all photosynthetic. Protozoa, they are unicellular always, and they lack tissues, but they share similarities in cell structure, nutrition, life cycle, and biochemistry under this term protozoa. And they have these very interesting, complex life cycles. So we see a lot of parasites that we call protozoa cycle through many different hosts, and they are infective to some and not others. Now, algae, always photosynthetic. There's unicellular, colonial, or filamentous forms, and they always contain chloroplast. They do have a cell wall, and sometimes you may see that they have flagella. Now, these guys are fascinating as far as the myriad of structures that they form. So these are what we call diatoms. Now, the most important group for recycling, producing oxygen in the environment, and reducing the amount of carbon dioxide are the plankton. So most of these you find within marine water. Most are free-living algae, and the plankton provide the basis of the food web in most aquatic environments. They're the largest producers of atmospheric oxygen. And the only downside of these are a group of algae, dinoflagellates, can cause red tide. So basically this is the result of eutrophication, where you have a buildup. There's a lot of nitrogen and phosphorus in the environment. And these algae can release toxins that when we consume the shellfish that's contaminated with the toxin, it can lead to food poisoning and potentially neurological symptoms. So this is the only link actually here with disease when we're talking about algae. So otherwise, consuming algae is non-pathogenic except for the case of intoxication with dinoflagellates. Now, as far as classifying algae, they're always classified according to the type of pigment, the cell wall, and they're used surprisingly for different cosmetics, also food and medical products as well. So there's a whole list here of, as far as the common name, different pigments, important. So here's even, we mentioned food, right? Here's a seaweed, Rhodophyta, that's a source of agar and actually a food additive. Another one, brown algae, is a source of an emulsifier. Now on to protozoa. These are again single cell, they're unicellular. Most are harmless and they need a moist habitat to thrive, but some of them exhibit this fascinating life cycle as far as the hosts that we see they can cycle through. And some of them also are spread via insect vectors. Now, for example, I'm going to give you guys in the next slide an example of a genus species of a protozoan that can cause disease in humans. But some protozoa have the ability to form similar to an endospore in bacteria. They can go dormant in the form of a cyst. So whenever they're in the active feeding stage, this is referred to as a trophozoite. This is the motile feeding stage. But what can happen here is that in an environment where it's desiccated, there's a lack of nutrients, it goes from the active trophozoite stage, the active feeding stage, to this formation of a cyst. So you have this early cyst wall formation, transition into a mature cyst. This is a dormant resting stage. Now the cyst form can actually be transmitted and then can go on to, once you actually contract or transmit this form of a pathogen, once the environment has been restored, so moisture has been restored, nutrients as well, it can then, similar to bacteria, it can germinate, the cyst wall breaks open, and the trophozoite is reactivated. Now, all protozoa also reproduce asexually via mitosis or multiple fission, and many also surprisingly reproduce sexually. It's this process called conjugation. Now, I would like us all to be aware that there are 4 basic groups of protozoa, and it's based on their movement, their reproduction, and also life cycle. So the 4 are Mastigophora, and there's pictures on the right here, always have flagella. So primarily flagellar motility, and they can undergo that conjugation that I just mentioned. Also, number 2, Sarcodina. These are primarily amoebas. Most are free-living, and you see they have these characteristic pseudopods, or these cytoplasmic extensions. Ciliophora, they always have cilia. They can form trophozoites and cysts. They're harmless, so they're non-pathogenic. Always, you usually see chlorophyll in the chloroplasts within Ciliophora. Finally, Apicomplexa. Typically, there's no motility except in the male gametes, both sexual and asexual reproduction, and they have these complex life cycles. So there are a ton that can actually cause disease here. In the case of the amoeboids, Entamoeba histolytica. Also, if you guys have heard of Naegleria fowleri, there's been a couple of cases of that in Connecticut, from actually inhaling water in a stream or a river. And this particular parasite can actually cross the blood-brain barrier and is fatal. So this is something that can result in a nasty infection. Also, Balantidium coli is another one that causes a disease. It's zoonosis in pigs. And Giardia, this is more of a common one, STD caused by Trichomonas vaginalis. These are all vector-borne, so Trypanosoma brucei, also leishmaniasis. We don't see these around here. They're more in the tropical geographical areas. Also malaria, so this is primarily in Africa, for example. There are millions of people that contract malaria. It's a huge problem. Vector-borne via mosquito. Toxoplasmosis, this is caused by infection with Toxoplasma gondii. This is one that can actually cycle through, interestingly, different hosts, starting with a rodent, a mouse, or a rat, and then cycle to a cat, and then it's shed in cat feces, and humans can contract this. So that whole crazy cat lady phrase actually has some scientific evidence behind it, because it does change the brain chemistry once it is infective. And also Cryptosporidium. So the protozoa that cause disease exhibit these interesting cycles. So a lot of them can be spread via insect vectors. So here's an example. Trypanosoma brucei causes African sleeping sickness, Chagas disease in South America. So neither of these we see around here. But they are spread via an insect, the reduvid bug or the kissing bug, which actually will shed the parasite in its feces, and then that parasite can enter a cut or a scrape on the surface of human skin, and it can also passage through other animal hosts as well. And then we have another parasitic protozoan, Entamoeba histolytica, which is an infective amoeba, and this causes dysentery, and this we see worldwide. So this is spread actually from eating contaminated food or water, and it's in the cyst form. So you actually have ingestion of the cyst, and then once that cyst enters the digestive system, the acids in the digestive system will actually trigger the release of trophozoites. And so there's this maturation or return back to the active state, and then through fecal exit, the cysts are shed once again so they can be transmitted via that route. Now, lastly, there are the helminths. So again, these are parasitic worms, and there's two groups of them, the flatworms and the roundworms. Flatworms, they have no definite body cavity. They do have this primordial digestive tract, a simple excretory and nervous system. And of the flatworms, there's tapeworms, which can be hundreds of feet long in a whale, for example, up to 6 feet long in humans, or flukes, which are non-segmented. So tapeworms have these segments called proglottids. Also, roundworms, also called nematodes. So these are round. They have more of a developed digestive tract and a protective surface cuticle. They also have spines and hooks on the mouth, but in this case, the excretory and nervous system usually are poorly developed. Now, as far as classifying the helminths, usually they're classified according to their shape, size, their organ development, whether they have these barbs, hooks, or other special structures, how they reproduce, the hosts that they cycle through. So all of these are used. Also, the appearance of eggs and larvae. And just a note that, so for the parasitic worms, so when they can actually cause disease in animals or humans, there are certain forms that are infective, and it's usually in the form of an egg or larva, so it's usually not in the form of the adult worm. So if you can see the squiggly worm, usually it's not as much of a risk as compared to eating, for example, food that has these more dormant type of the form of the worm, like an egg or a larva, which then could develop into the adult in the digestive tract. Now, just some fun facts. There are approximately 50 species that are parasitic, and they're all over the place. There are some that we don't see around here, and we will be talking more specifically about those that are unique to the tropical areas in chapter 23. But as far as how you would actually contract one of these parasitic helminths, like I said, it's from the larva, their eggs, from eating contaminated food, also from soil. So some of these can actually burrow, the larva can burrow through the skin and travel through the bloodstream to the lungs, or even from the digestive tract into the bloodstream and then to the lungs. They can also be transmitted via contaminated water, and then, like we said, some, along with other parasites like protozoa, can be transmitted via insect vectors. And these types of diseases afflict billions of people.